Can everyone hear me? So welcome everyone to the second round of this really exciting meeting so far on actually looking at NAMs in the global landscape. So I have the privilege of saying a few words. So I'm going to tell you about the program at the US FDA, because we're not sitting back waiting for you to come to us with NAMs. We are out there actively looking for partners to work with us and with our NCTR laboratory to develop hand in hand with our regulators methods that then we can put into regulatory use and then actually maybe put them into ILMIRAC, which was 20 countries, food regulators, that we met on Monday. So what we're doing is, Christy mentioned a little bit our alternative methods work group. That's across the agency, senior regulators from each of our programs. We have to report to the commissioner every month how our qualification programs are coming. So there's a lot of pressure on us, good pressure, to really work on that together. And coming to a meeting like this and seeing all the exciting science, and I'm always looking for groups to partner with. We also have a program that our Alternative Methods Work Group runs where we invite researchers that have methods with a context of use to speak to the FDA and introduce your technology, because we want to make sure that our regulators don't see a technology for the first time in an application, that they're very comfortable for all of this new technology when they see it in some application for a drug, device, food, vet drug, etc. Biologic. And so it's a program that you could, if you go to alternatives@fda.gov, and well, that's our website. If you type into Google FDA alternatives, you'll get to our website. You can reach us with our alternatives@fda email address, and I thought we would be, like flooded with emails, but I mean, I was telling someone, I get more from Amazon than we get from people out here, so please feel comfortable to talk to us. We have a wonderful partnership with NCTR where we regulators ask them questions, throw up our hands and say, how can we answer it? And then together we answer the questions. And I'll just make one example and then I'll turn it over to Sonia. The first question that you ask in cosmetics is, what's dermally absorbed? So we have Louisa from NCTR is working with our cosmetic regulators to talk about models that they can use then in a regulatory assessment, figuring that whatever we find from her models, that which are the best models to use, and then what type of approach can we take when we see how much absorbed. So TTC approach, full systemic approach, so it's really, she meets monthly with regulators to develop what they need. And I think that's a good program that we'd like to see happen more at, with NCTR. We do that already, but more with our stakeholders out here. So with that, I will turn it over to our first speaker, was Sonia Becken, a good colleague of FDA, also a good friend of mine. I'm very disappointed she's not here in person. She chairs the 3Rs working group at EMA, and she's been a great partner with all of us here. So are you online, Sonia? Ah, there you are. Hi. So much. Yeah, thank you for speaking. I wish you were here in person. So it's your floor.
Thanks a lot, Susie, for this very kind introduction, and really apologies that I could not join you today, but I'm hoping also to pass the message from the European regulatory perspective that the focus is truly on NAMs. So I hope you can see my slides. Can you please confirm? Yeah, thank you. So thanks to the organizers for giving me this opportunity here today to present the 3Rs implementation at the European Medicines Agency, what's in the past, the present, and the future, of course, because that's really important, and that's what we're discussing here today. So yes, there we are. So this is a bit the outline of my talk. I will be presenting to you what has been EMA's commitment to the 3Rs, so a bit of historical perspective. I will not go as far back in time as our Japanese colleagues this morning, but I'll give you some insights. Some information on the new 3Rs working party and its work plan, and then of course some take-home messages, and also shout out to the method developers and end users out there. So EMA EMA has been truly committed with regards to the 3Rs, and as exemplified here by a statement published on the website in 2011 saying that the agency commits to the application of replacement, reduction, and refinement of animal testing as detailed in Directive 2010/63 of the European Union, and that to this end, a joint expert group on 3Rs had been created to promote the best practices in the implementation of 3Rs in the testing of both human medicines and veterinary medicines, which is under the remit of the European Medicines Agency. So, what about this joint expert group on 3Rs? This was a group set up in 2010, and you can see that we had a dedicated web page at the time of this JAC 3Rs, followed a few years later by a joint 3Rs working group. And on this web page, you could find all the information related to the 3Rs principles and the EMA role regarding that. Also, all the actions that had been taken by these JAC 3Rs and Joint 3Rs working group, and all the scientific guidelines pertaining to fostering regulatory acceptance of NAMs. Now, a bit of insight on what this JAC 3Rs and Joint 3Rs working group accomplished over this 7 years period. Basically, these two groups focused a lot on setting up a real regulatory framework that fostered uptake of 3Rs testing approaches. So really the groundwork, how can we achieve regulatory acceptance of a novel approach methodology, a NAM, or 3Rs method? So we had a guideline on basic principles of regulatory acceptance of NAMs or 3Rs for testing of human and veterinary medicines. That was drafted and published on the website, and which gives you a high-level insight on the principles for regulatory acceptance. Moreover, there were specific guidances pertaining to quality control methods and also an extended review of all EMA and VICH and ICH guidelines, where the opportunities for implementing 3Rs and the already implemented 3Rs opportunity were detailed. These overviews are now being revised currently because both, of course, date back from 2017. We had an important work stream on batch release testing, since that area consumes most of the animals still in the testing of human and veterinary drugs. And we established close collaborations with European organizations such as EDQM, which hosts the Pharmacopoeia, the European Reference Laboratory for the Validation of Alternative Methods, and other EU agencies, and of course pertinent international consortia and organizations in the field. All of the achievements were published in an overview which dates back from 2016-17 and which you can access here. Now, looking to today, and let's have a look at the EMA's regulatory science strategy, which contains really recommendations that are dedicated to the implementation and fostering of 3Rs or NAMs, and to achieve swifter regulatory acceptance of those. And to get there, of course, we need to discuss criteria for regulatory acceptance. So really delineate what is understood under qualification, what is understood under context of use, what are endpoints of reference, and what could be reference compounds that we could test the methods with. And to draft those criteria for regulatory acceptance, it is recognized that engagement with relevant stakeholders is, of course, of key importance. And also in this regulatory science strategy, there was the mention to set up a 3Rs working party to foster all this work. And indeed, in November of last year, a strategic and visible working party was set up that will really monitor and supervise all EMA's 3Rs activities. And since these are multidisciplinary by nature, we are covering human and veterinary drugs, covering the areas going from quality control methods and batch release over non-clinical testing of human and vet drugs up to efficacy testing in clinical studies for veterinary drugs. We have also a composition of a core group that reflects this multidisciplinarity. Of course, it's only a restricted group, so in its work, the 3Rs working party is supported by specific operational expert groups, such as a batch release testing group and drafting group that will help us in drafting guidelines, reflection papers, and so on. Moreover, specific non-clinical and new approach methodologies European specialized expert community has been set up, and this encompasses both assessors from the regulatory network in the EU as well as academic experts that will really assist the EMA and the 3Rs working party and the non-clinical domain in its work plan actions. Moreover, we have the full support of the Scientific and Administrative Secretariat, of course, at the EMA, and we have again our observers on board, which are the European Commission services, EGFAM and ETQM. Now, what is in the work plan? We have different guideline activities, again, because this is really, again, what is coming out and what is helping applicants and end users to foster regulatory acceptance of their novel methods. First of all, we'll revise our guidelines on the principles of regulatory acceptance of 3R testing approaches with the aim to include a specific definition of critical 3Rs related terminology. Now, what is understood by that? We hear in several venues that there is really a misunderstanding related to terms like qualification, context of use, fit for purpose, and so on. So the intention would be to really enshrine into this guideline what we understand under these terminologies and who are the actors involved. Moreover, we'll add annexes that will provide specific criteria for the acceptance of microphysiological systems, including organ-on-chip technologies that are being used for specific contexts of use for testing human or veterinary medicines. And these will be annexes with the purpose of having flexible recommendations that can be adapted in view of the state of the art and also by learning from experience. Moreover, we'll draft a new reflection paper on the alternatives to the use of non-human primates, and we'll revise our reflection papers that already provide an overview of the regulatory testing domain and opportunities for 3Rs implementation. And this exercise is ongoing as we speak. Now, of course, we are talking about NAMs today and about regulatory acceptance of NAMs. And the whole work plan of the 3Rs working party is really constructed around the ultimate aim to get these NAMs into our dossiers, into our regulatory applications, and also into our guidelines. And when we're discussing uptake, we can see that there is a gradient of activities going from the development of context of use-based qualification criteria up to the actual qualification of the NAMs. So what will we do as activities to accompany this? We'll organize multi-stakeholder workshops on MPS and organ-on-chip models for specific context of use with the aim of defining these regulatory acceptance criteria or qualification criteria. Moreover, we realize that we can't do this alone. We really need to collaborate on an international scale to achieve harmonization of those criteria and to foster uptake more swiftly across the globe. Of course, we will need some collaboration in the area of modeling and simulation, since a lot of these models are accompanied by those type of approaches. And we'll have specific actions related also to updated guidelines, such as the ICHS-5 related to reproductive tox testing with its specific qualification criteria that are proposed there for alternative methods for embryo-fetal development testing. We'll also look at the cardiovascular safety pharmacology space with the best practices and general principles for in vitro testing methods that are proposed there. And we'll have a work stream on skin sensitization testing as well. And finally, and not unimportantly, we will support our innovation task force at the EMA and the scientific advice procedure for the actual qualification of methods. Now, let me just introduce the Innovation Task Force for those that are not really acquainted with that. The EMA's Innovation Task Force is in fact a dedicated forum for early dialogue with the regulators and other stakeholders involved in the field. And we're talking here about NAMs and 3Rs, so these are small and medium-sized enterprises, researchers within the academia, but also bigger public-private funded consortia and the pharma industry. And the focus is really to foster this regulatory acceptance of NAMs, not only the qualification, but also encourage the further development of those. Now, the meetings are really an informal exchange of information, and during the meetings, some early, non-legally binding guidance is provided to method developers or end users to really assist them in the further pathway towards regulatory acceptance or qualification of their methods. The best available expertise is selected, of course, from the agency network, from the EMA working parties and committees, and people will assist to these meetings and provide their input from their expertise. And not unimportantly, these meetings are, of course, free of charge, and we really reach out to all of those to engage into this first dialogue with the regulatory network and promote uptake of their methods. Sorry. Now, other activities are also on the work plan, and there is, of course, again, a big work stream on batch release testing, but also we have a rather important focus on multi-stakeholder interactions. And we engage into this interaction through an annual stakeholder meeting with the 3Rs Working Party. And moreover, we are in an exercise of mapping the cooperation with all our EU and international NAM and 3Rs stakeholders. And not unimportantly, in the space of NAMs, and you heard it also already from Susie, we need to get our assessors acquainted with all these novel methods so that when they see them in an application dossier, that they are not surprised. So we focus a lot on training of 3Rs methods and discussing best practices among the members of the network. Now, beyond 2023, we will also focus on the organization of a larger conference to introduce the work and the roadmap for 3Rs at the EMA. We'll also review the methods that are most promising to be included in a qualification program, and we'll work on a database together with our colleagues at EGVAM and EDQM. Now, an idea about how the annual stakeholder meetings go about. We had our first meeting in February of this year, and we had 4 big stakeholder groups that were invited to it. We had the industry and the CROs on the one hand side, the EU agencies and the organizations of the EU. We had a group with NGOs and animal welfare associations, and of course we had the research consortia and other stakeholders. Now, on the agenda, we had a first live broadcasted introductory public session, followed by an interactive Slido to query the public on particular points related to the work plan of the 3Rs Working Party. And this public session was followed by closed sessions per stakeholder groups. And all the topics which were on the agenda were proposed by the stakeholders. Now, we felt that all the discussions we had during that very long day were quite constructive, and the emphasis was clearly on the current work plan actions, although a number of stakeholders presented also additional topics that will be considered for future actions. And you can find the public session report there on the QR code. Now, just an idea about the Slido. We queried the public on what they think is the most important aspect when they were thinking about 3Rs in regulatory testing and drug development. And you can see really at the heart of this word cloud, the words acceptance, validation, regulatory acceptance and testing. So this is also what we will focus on. And this translates also in the Slido proposed prioritization of the work plan actions of the 3Rs Working Party, with on top clearly the work stream on qualification of NAMs. Now, just an idea also on the reform of the EU pharma legislation and the aspects on 3Rs. So clear in April of this year, a draft new directive and regulation for the human pharmaceuticals was published. And in this, both in the recital, so in the introductory statements, as well as in the articles, there are important 3R statements. One of them, of course, is the reference to the Directive 2010-63 and its 3Rs principle, which is clearly harnessed now in this new legislation. Moreover, there is the mention of avoidance of unnecessary duplication of testing using live animals, with the recommendation to set in place procedures to facilitate not only joint animal testing, but also the reuse of animal test results. And also to make it clear that a non-clinical test was defined as being not only in vivo testing, but also in vitro testing, in silico, in chemical testing, although this was already understood previously in the old legislation. Here it is spelled out quite exhaustively. Moreover, there is in the articles the requirement now for marketing authorization applicants to demonstrate that the principle of the 3Rs of animal testing for scientific purposes has been applied in compliance with the directive. And moreover, that animal tests shall not be done in case a scientifically satisfactory non-animal testing method is available. Finally, the responsibilities of the EMA with regards to animal use and the 3Rs are clearly listed, and this relates also to the work plan of the 3Rs working party. Now, of course, this is not yet a definitive version. The road to implementation is still quite long. It will take a number of years, but still it's really promising that these far-reaching 3Rs aspects are taken into account. I would like to finalize with some take-home messages. So I hope I could demonstrate to you that EMA is also clearly committed to the 3Rs, and we are really inviting all end users, method developers to come to us and present their novel methods. And of course, we are also actively reaching out to those and reaching out to the consortia and the academics and the end users to increase the collaboration with the EMA to foster uptake of NAMs. 3Rs Working Party is now an official 3Rs hub at the EMA. We have a 3R strategy in place, an ambitious work plan that will hopefully foster swift uptake of NAMs when they are far enough in the qualification or when they are deemed acceptable for application in the regulatory framework. We want to do this through the engagement and open dialogue with interested 3R stakeholders. And one of the essential tools for this is the Innovation Task Force on 3Rs. And finally, we also want to remind that global regulatory collaboration is key to achieve progress in this field, which is, of course, global by nature. And we'll focus on the next work is really the regulatory acceptance of NAMs through the development of context of use-based qualification criteria. Although this is work in progress, we can see it from the actions throughout the scientific community and the regulatory community that is high on the agenda. And I hope we can really collaborate in this space to achieve progress sooner than later. And with this, I would like to thank you for your attention, and I'll take any questions.
Do we have time for a question, you think?
Yeah, a bit, maybe one or two.
One question. Okay,
Maybe just one question. Bill?
Just one, yeah.
Well, thank you very much. Very clear presentation. I'm just thinking about, contacts and exchange. Some of the principles you mentioned about number of animals used in a study, what is appropriate animal model, how should those models be used, those are already being reviewed on each regulatory campus within the United States by the Animal Care and Use Committees that are enforced by OLA, which is an NIH sponsored organization. It's the NIH Office of Laboratory Animal Welfare. So it might be good to coordinate with them, since they're already doing similar kind of work, and just to crosstalk a bit about how they're reinforcing and enforcing this. These are very serious commandments on how you handle animals and use animals, and of course, in order to get something published, you're supposed to guarantee that you follow those guidelines. And those committees are very rigorous and very strict. So I'm just saying it would be helpful to make sure you're bridging the gap to existing use of animals.
Yeah, of course. And here also in this perspective, we are collaborating and we are discussing with our colleagues also on the European side of the animal ethical committees and all the work that is done by DG Environment, because they are reviewing also per project the number of animals and the application of NAMs. But as mentioned here in the reform of the legislation, it's more related to the whole program for drug development, and where we have, as the regulators, more an umbrella view over the complete use within the drug development program, rather than as a per procedure view, as is often the case in the ethical committee. So there is, of course, need for crosstalk. I totally agree with you.
Okay.
Okay.
I think with that, we shall move on to the next speaker, which I will introduce. So the next speaker, we have Dr. James Chan. So he's a principal investigator from the A-STAR Institute, the Singapore Institute of Food and Biotechnology Innovation, and also the A-STAR Skin Research Laboratory. He's currently an investigator there with extensive experience in PBPK modeling. And he has done extensive work in the modeling of half-life of environmental contaminants, such as PFAS, and also prediction of biokinetics for pharmaceuticals, nutritional and consumer care compounds. So without further ado, let us invite Dr. James Chan to the floor. Thank you, James. Yeah.
Hi, good afternoon, everyone, and thanks for the invitation to speak. So it's my pleasure to share with you a little bit about our work in toxicokinetic modeling. Today I will be talking about a more specific set of chemicals, which are the PFAS chemicals. So very often when I mention toxicokinetic modeling, it's a bit of an abstract topic, right? So I thought I'd start off by giving you a mental image about how you can think about toxicokinetic modeling. And it looks a bit like this, right? So as a NAM, you can think of it as like a digital twin or a virtual human subject, and that's what I like to call it. But of course, it doesn't look as nice as this, right? It looks a little bit more mundane, right? This is actually what it looks like. It's a mathematical model. Essentially, if you look at the top left panel, we have what we call a compartmental model, where we just describe mass transfer, right? Substances can enter the body and then distribute around the body through blood flow. They can enter into organs, they can leave organs. All of it is governed by blood flow. And it's essentially an effort to describe how much of a substance is found in the bloodstream, in different organs, how long they reside there, and how are they eliminated after they enter the body. Right, so this is basically the skeleton of the model. But all in silico models need to be parameterized, right? So the middle section tells us how we parameterize them, right? We combine data from in vitro digestion models, and it tells us how much of that substance will get released during the digestive process. We also have various assays which we run, which tells us a little bit of the whole picture, how much gets absorbed, how much gets distributed, metabolized, excreted, and so on. And ultimately, what we like to end up with is on the right side, right, where we try to predict both plasma and tissue concentrations. Plasma is often done, but tissue is relatively less common, right? But we focus on tissue because the tissue levels are actually the site of action. Whether we're talking about efficacy or we're talking about safety, that is where the compound is exerting its effect, right? We often assume that plasma and tissue levels are interchangeable, but there are many instances where they are not, right? Due to transporters that might concentrate or deplete that organ of that particular chemical entity. Right, so having given you a little bit of background of what toxicokinetic modeling is, I'm going to introduce you to this group of notorious chemicals called PFAS, which we also call the forever chemicals. Right, if you don't already know, you should know by the end of my talk. This is a superfamily of chemicals. There's more than 14,000 at last count, and they are found in almost all consumer products, right? Because they are oil resistant, they are heat and chemical resistant, so they've been used for industrial and consumer applications for many decades. We also find them ubiquitously in the environment. They are found in our food chain. They're also found in humans. Why we are concerned about them is because they are very resistant to biological degradation. And I'll describe this more later. We are exposed to them through the products we use, through the food that we eat, through the packaging that we use, through the air that we inhale, through contaminated food and water, right? So they are coming into us from almost every possible absorption route. We are concerned with them because some of them have a very, very long half-life, right? So they can be as long as up to 7 years, right? And this therefore opens the opportunity for bioaccumulation, right? Because we take them in a lot faster than we can eliminate them. They have also been associated with various safety concerns, including thyroid abnormalities, liver, kidney, immune, neurodevelopment, and even fertility, right? So shown on the the right side is a structure of a prototypical PFAS, which we call perfluorooctanoic acid, or PFOA for short. And in fact, it's one of the building blocks used to make Teflon, or non-stick coating, which we encounter very often in our cookware. So right off the bat, when we looked at this set of compounds, we realized that there's a lot of disagreement in terms of the half-life of PFOA, right? Beginning from animal studies, depending on which species you are studying them in, they can be as short as several hours or even several months in monkeys, right? So there's very poor interspecies correlation. Then on the human end, even in the human studies, they don't agree, right? So in the middle panel, I'm showing you data from a United States patent application. This was created by 3M, where they actually administered PFOA to cancer patients, right, at a very, very high dose of 50 milligrams, right? We never get exposed to 50 milligrams in our entire lifetimes, right? So, but these subjects received this dose, and they estimated a half-life at about 116 days, right? So it's about 4 months. Now, on the right-hand side, you have the other extreme, right? So biomonitoring studies have estimated the half-life to be within 1.3 to about 4 years. So this is really very different, right? So even when you have clinical data from humans, they don't necessarily agree. So we looked at this problem, and we decided to approach it by looking at a number of different PFAS compounds, right? So we picked PFAS that have very short half-lives and PFAS that have very long half-lives. The goal was to try and understand why are these different, and can we explain why some PFAS have such long half-lives, right? There is a certain relationship, apparent relationship with the carbon chain length. So if you look at the top of the table, you have 4 carbon chain length PFAS that have half-life of several days, and to the very end, you have 6 to 8 carbon lengths which have half-life of years, right? So there's a bit of relationship there, but this doesn't fully explain everything, right? So we set out to try and identify, to try and answer 2 questions. First is, why do biomonitoring and clinical studies disagree so vastly on the half-life? And can we identify PFAS that have this kind of long half-lives for risk prioritization? So I'm going to walk you through now a bit of a structure-activity relationship, right? So this is the structure of PFOA. If you look at the structure, immediately you realize that first of all, it's an acid, right? It's an acid with a dissociation constant of 3.8. What this means is that in the blood, it will be almost completely fully ionized, and that makes it unable to cross biological membranes by passive diffusion, right? The second very interesting thing is that you can see it's decorated with fluorines, right, instead of hydrogen. So this is a fully fluorinated molecule. In fact, if you think about it, it looks like a fatty acid, except that this fatty acid is not filled with hydrogens, it's filled with fluorine atoms, right? And because it is ionized, it cannot cross biological membranes, we are dependent on transporters to move it around in the body, right? So at the time when we started investigating this, all those transporters that I boxed up in red are the known transporters found in the intestines, kidney, and the liver. And so we thought that PFAS would only be confined to these organs. right? So we set out to model PFAS, and I won't go through all the technical details, but we essentially ran a whole battery of assays, right? It takes about 12 to 13 assays to characterize one PFAS, right? So it's a pretty large amount of work, but we ran the assays to parameterize the model, and then we use the in silico model to try and predict plasma levels and also understand the half-life. All right, so let me first walk you through what we found when we looked at the clinical study. So as I mentioned, 3M ran this study, administered a 50 milligram single dose, and they actually had plasma concentrations data for this particular chemical, right? So we were able to recapitulate all the data points that were reported in the study, from the Cmax to the AUC and also the half-life. So at the bottom line of this table, you can see that we were very close to the predicted half-life, right? Almost a one-to-one match. So we can recapitulate the half-life in a clinical study. We also looked at another scenario where in real life, we don't get 50 milligrams single exposure of PFOA. Instead, we have very low but sustained chronic exposure to PFOA. So take, for example, 70 nanograms per day, right? So we ran this in our model, and we were also able to recapitulate the long half-life that's been observed. The question is this: why are the clinical and biomonitoring estimates so different, right? So this is a very interesting question that we set out to tackle. So as I mentioned, transporters are very important, and based on what we knew, I'm using an example of a kidney here. So this is a kidney cell, and on the left-hand side of the kidney cell is basically entry of transporters that control the entry of PFAS into the kidney cell from blood. And on the right-hand side are transporters that reabsorb it from the urine into the kidney cell. As you can see, transporters are bringing in PFAS into the kidney cell, but there's no way for the PFAS to leave the kidney cell. Now, if this was the case, we would expect massive amounts of accumulation of PFAS, right, in the kidney cell, but we obviously don't see this. So there has to be a release valve, right? There has to be a transporter that will actually pump it back out into the blood. So we added this transporter into the model, and we were able to then harmonize or explain both scenarios, right? So in the clinical study run by 3M, it was a very high dose, and this transporter can be saturated, right? As a result of this saturation, right, we cannot reabsorb PFAS from the urine, right? So that's why more of it can be excreted in that high dose study. But when we talk about the low dose exposure, which is a more realistic setting, at this low dose, this transporter is not saturated. So it continues to reabsorb PFAS, and that is why we have this very persistent presence of this compound in our body, right? So this was a new discovery that allowed us to actually explain both scenarios, and actually continues to reaffirm biomonitoring estimates that the half-life is really years instead of months, as was reported by the manufacturer. Right, so how does our model estimates compare with other estimates done by other regulatory agencies? So there was a forward dosimetry study run by Health Canada, and we are reasonably close, right? Based on the total diet study, 70 nanograms per day intake, you have about 2.5 nanograms per ml estimated by them, and ours was 7.8, roughly around the same fold change. And the reverse dosimetry estimated by EFSA, to achieve a serum level of 2 nanograms per ml of PFOA, an intake of about 0.18 nanograms per kilogram was needed. We estimated about 0.28 nanograms per kilogram, so reasonably close with the other estimates that were reported. Now, another very interesting thing that we discovered was that we found a new transporter that transported PFOA. This transporter has never been reported, right? But it makes sense because this transporter is called MCT1, right? It stands for monocarboxylic acid transporter 1. And it actually transports both short and medium chain fatty acids. So, for example, it transports octanoic acid, which is one of the nutrients that we have in our body. But as I said, because PFOA looks a lot like a fatty acid, right? So it also transports PFOA. The important thing to note is that this transporter is found everywhere in all organs of the body, right? So this means that you can find PFOA being distributed by this transporter into all body tissues. And therefore, it's no longer confined to just the liver, the kidney, or the intestines as we once thought, right? So this explains why you actually see PFOA in every organ in cadaver studies, and you actually have such broad tissue effects when it comes to the safety. And it also increases the volume of distribution. So when a compound is very broadly distributed, it's very hard to remove it from the body. So therefore, it increases the half-life as well. All right, so having done a very thorough study on one PFAS, we decided to repeat this whole process on the rest of the other four PFAS that we had shortlisted, right? So I'm just going to give you a quick rundown. This is all the data that we generated. Took 3 full manpowers, 3 years to conduct this whole study, but I'm just going to give you a 30-second snapshot of it, right? So on the right-hand side, we have all our PFAS that we studied, and then we predicted the half-lives. They were reasonably close to biomonitoring estimates, right? But more importantly, what we learned was that what controls the half-life is basically how much gets reabsorbed by the transporters. So instead of looking at 8 different transporters, now we narrowed it down to just 2 key transporters, which I have boxed up in red. These are the ones that control its reabsorption, and the more it gets reabsorbed, the greater the half-life. The other key feature is that compounds or PFAS that have very high protein binding also have very high half-life. Because binding to proteins in plasma means that they cannot get filtered by the kidney, right? Because it's like a magnet, it keeps it in the bloodstream, right? So altogether, they contribute to the very high reabsorption of these compounds that exhibit high transport and also high protein binding. And of course, those that are substrates or MCT1, which are the long chain, the long half-lives, also have greater distribution, right? And therefore, it contributes to their long half-life. So in summary, what we found was that we found a number of assays that we can now develop to screen for compounds that have long half-life, right? PFAS that have very high half-life, and we translate this into a risk prioritization strategy. So there's more than 14,000 PFAS out there. You can't possibly study them all, but you can potentially rank them in order of priority, right? So as I mentioned, it's basically protein binding and transport by certain key transporters. So if the compound has very high protein binding, so protein binding of 95% or below, we consider low risk. Anything that's 95 to 99% and above is considered moderate risk. Those that are transported by the transporters I mentioned that have reabsorption are also moderate risk. If you want to stratify them further, we can look at how much is being transported. And so we have a cutoff of about 10 microliters per mil per milligram protein. This is just a unit where we measure transport rates. So anything that's less than 10 is considered moderate risk. Anything that's more than 10, potentially high risk. And also if it's transported by this very unique transporter, MCT1, altogether you can consider this to be of high risk and what we should prioritize for full model building. Right, so this is how we basically use modeling to identify mechanisms and translate these mechanisms for risk assessment. So to conclude, I just want to share that these models help us obtain mechanistic insights that cannot be easily obtained from regular human or clinical studies, right? And the main organ that controls PFAS removal is really the kidney. And therefore, if we want to design future PFAS, we should have certain properties in mind to reduce the protein binding, to reduce the affinity to transporters, right? So this has been published in part in this article, and we are continuing to work on a second publication. I thank the people who were involved in this work and happy to take any questions.
We'd like to open the questions to the floor. Any questions? Yes, Gonzalo.
Thank you. Unusually clear explanation of what these models entail. I'm curious, I mean, one thing is the modeling that you do, but another thing is obviously then real-life data. So did you have the opportunity to actually quantify residues of perfluorooctanoic acid, for example, in the multiple organs? And I'm specifically interested in the fat compartment. What did you find there?
Yeah, so what we do have is data from cadaver studies. So we have some reference there in terms of PFOA levels in fat. We can also predict them because they even get distributed to fat. In fact, they are like fatty acids. So we have these 2 data points, a measurement and also a prediction from the models. And although I didn't specifically check for it in fat, we do have those levels. In fact, we can extract levels from all the different body tissues and then give a sense of how much penetration there has been for these PFAS into each of these tissues.
So the reason why I'm asking is that I'm expecting that they will partition quite efficiently to the fat compartment, and that may constitute a long-term deposit where perhaps there's less complexities associated with transportation back and forth. I mean, it may just lodge there. So, but anyway, I mean, congratulations, really clear explanation of the work that you've done. Thank you.
Thank you.
Any, yes.
So the intercellular distribution, did you look at that as well? Because they will likely be stored, of course, in vesicles, lipids. And would there be access then to the targets where they might have an effect on the biology?
Yeah, thanks. That's a good question. We didn't specifically look at that scale, so our modeling was mostly at the tissue level scale. But yes, certainly once it's uptaken into the cells, they could be sequestered, just like any other fatty acid. So wherever fatty acids go, you can think of it as that's where these compounds would go as well.
Yes, we have another one.
Hi. One of the benefits of PBTK modeling is cross-species extrapolation. So I was wondering if you've looked at other species and if the transporter differences in the species explain some of the differences that you saw.
Yeah, thanks. We didn't personally do cross-species extrapolation, but we did look at how other people model in different animal species. And yes, the biology of the transporter expression is very different, especially in rodents. So they don't express the transporters that reabsorb. They only express the transporters that secrete. And that's why the half-life in the rodents are so much shorter than in humans. So in humans, we just somehow love to retain these compounds. And that's basically the driving force for the species difference.
Any other further questions for James? Okay, if not, James, I have one question. It's wonderful that you share with us such a model. I think it's extremely useful. And what are some of the limitations of this model? If you could, share with us a bit more. Yeah.
Yep. So in the models like this, they need to be explicitly described, right? So we can't model beyond what we know. So we have to know, or we have to look at all the possible mechanisms. If we miss the mechanism, then that can render the model less accurate than we want it to be. So it's only as good as what we know, and that's basically a constraint. So it's quite different from machine learning approaches where it's a data-driven approach, right? Ours is a mechanism-driven approach. So we have to understand and characterize the mechanisms as thoroughly as we can.
Okay. Thanks. Any last questions from the audience? No? Okay. Thank you, James. Thanks.
Thank you.
Okay, our next speaker, we have Dr. Yoko Hirobayashi. She's the director of the Center for Biological Safety Research at NIHS. And Dr. Hirabayashi oversees all the testing and research activities related to, comprehensive safety evaluation of chemicals, food, drugs, using various biological resources. She also chairs the steering committee of the Japan Center for Alternative Test Methods and Validations. And also, she's a member of the International Cooperation on Alternative Test Methods. And not only that, she's also the Japan National Coordinator for the OECD Test Guideline Programs for the Human Health Program from 2020, and also a Vice Chair from 2021. And with that, may I invite Dr. Yoko to give us the presentation. Dr. Yoko-san, please.
Thank you. First of all, I'd like to thank organizers, EFSA people, Dr. Kass, chairperson, for giving me an opportunity to talk about current work. So let me first introduce overview of the Japanese Center for the Validation of Alternative Methods, JACVAM. Then I'd like to talk about JACVAM's mission, organization, and past efforts. Finally, I'll show you our current activities towards the development of new approach method, NAMS. So, National Institute of Health Sciences, NIHS, consists of 20 research divisions, as already introduced by Dr. Honma, director of NIHS. Among these 20 divisions, the Center for Biological Safety and Research, CBSR, currently consists of 5 research divisions and 2 facilities. That is Experimental Animal Facility and Japanese Center for the Validation of Alternative Method, JACVAM for short. JACVAM was established in 2005 to promote the use of alternative methods to animal testing in regulatory studies. The photo on the left shows people who have been involved in the management of JACBAM, including the past directors of NIHS and the directors of CBSR, as well as former executive director of JACBAM, Dr. Kojima, who have supported JACBAM for many years, and current executive director of JACBAM, Dr. Ashikaga. On the right side of the slide, you'll see a list of organizations that are involved in international cooperation with JACBAM. In particular, JACBAM is a member of International Cooperation on Alternative Test Method, ICATM. We hope to further strengthen our cooperation relationship with the participating member of ICATM and celebrate our 20th anniversary in 2025. International Cooperation of Alternative Test Method, ICATM, is a voluntary international cooperation of national organizations established in 2009. Before the pandemic, ICATM have been meeting once a year to collaborate and share information on the latest developments in alternative methods and activities in their respective national organizations. ICATM just celebrated their 10th anniversary in 2019, and although the pandemic interrupted our face-to-face meeting, we met again at the World Congress for Alternative Methods in this August. And it is not yet indicated here, but UK became a new member separately from EU. Next, I'd like to talk about JACBAM's mission, organization, and past efforts. This is the web page of JACBAM, and URL is indicated here. JACBAM's policy and mission is to promote the use of alternatives to animal testing whenever possible, and to replace, reduce, and refine the use of animals while fulfilling the CBSR's responsibility to ensure the protection of the general public by evaluating the safety of chemicals and other substances in accordance with NIHS regulations. Here is the composition of the steering committee of JACBAM. The steering committee consists of members from NIHS, including all 5 divisions of the CBSR, as well as from the Ministry of Health, Labor, Welfare, MHLW, the National Institute of Infectious Diseases, NIID, and Pharmaceutical and Medical Devices Agency, PMDA. And this steering committee is chaired by the director of CBSR, and that is me. In addition, the advisory board, consisting of members from related industries, academic scientists, and student groups, have been established to provide high-level advice on operational policies. This is the outline flowchart to indicate JACBAM's activity in accordance with the policy and mission. In the test guideline validation process, JACBAM contributes to the officialization of alternative test methods developed internationally or domestically as OECD test methods. In particular, for alternative test methods developed in Japan, we assist developers in conducting validation. In regulatory acceptance process, JACBAM also reviews the acceptability of established OECD test methods to the Japanese regulatory authorities and proposed test methods that are considered appropriate. The following slide shows the structure in each of these processes. First, regarding the regulatory acceptance process, the steering committee constitutes an editorial committee to request the preparation of an evaluation report on the guidelines and other documents established by the OECD that are deemed to require more detailed explanation when conducting the test. The Regulatory Acceptance Board will then check the scientific validity of the test method, its acceptance by society, and its usefulness as an administrative test based on the evaluation report prepared by the editorial committee and report back to the steering committee. For this activity, the steering committee also receives requests and opinions from the Stakeholder Council, which consists of the broader range of related organizations than the members participating in the Advisory Council. In this context of the report is deemed sufficiently supportive for administrative acceptance, the committee proposed this to the MHLW. To date, we have proposed 40 methods, each of which has been utilized for regulatory purposes. Next, regarding the test guideline validation process, based on the proposal from the developer, the steering committee will propose the standard project submission form, SPSF, to the OECD, and simultaneously, the management team for the implementation of the validation will be formed. Then a peer review panel will be formed to review the result of the validation. A report is submitted to the OECD for adoption as guidelines. International cooperation is essential at each step of this process, and we work with the support of ICATM and other organizations. As of 2022, the OECD has a total of 75 guidelines for human health testing methods. Of these, 30 or 40% are guidelines that do not involve the use of test animals. Most of them are related to the evaluation of local toxicity, and in some categories, most of them are guidelines that do not use test animals. JAGBAM has contributed to the establishment of all guidelines related to human health. In particular, as mentioned above, we have worked with Japanese test method developers to have their methods adopted as OECD guidelines, including their validation, as shown in red. For the last part of my talk, I'll show you our current activities towards the development of new approach methods, NAMs. The development of alternative test methods that do not use test animals have been underway for some time, but with the advancement of science and technology and the spread of the philosophy of animal welfare, there is a need to develop new testing methods that include a combination of multiple evaluation methods as new approach method, NAMs, bearing in mind that limitation of evaluation by a single in vitro test method. The word NAMs in itself has a slightly different meaning depending on who uses it, but the US EPA definition is used here. For future NAM development, JAGBAM will focus on the development of test methods that contribute to the evaluation of systemic toxicity rather than local toxicity. The main focuses are reproductive and developmental toxicity, immunotoxicity, and toxicokinetic analysis using the microphysiological system, MPS model. First, let me talk about reproductive and developmental toxicity. We are contributing to develop methods using human iPS cells and zebrafish eggs. Of these, for the hand-one-look EST assay on the far right, the developers prepared a detailed review paper, DRP, with support from Jack Baum. Since this method using human iPS cells was very new to the OECD guidelines, DRP was required prior to the guideline development process. And unfortunately, it was withdrawn from the guideline due to the developers' reasons and the fact that it took too long time to prepare DRP. The recently published ICH-S5R3 guidelines for non-clinical safety evaluation for pharmaceuticals specify the requirement for non-animal test methods. In particular, many of the novel modality therapeutics that have been actively developed in recent years have high target specificity and consequently high species specificity, which in many cases make safety assessment using laboratory animals impossible, not only for reproductive toxicity. In addition, the non-human primate required for the evaluation of such drugs have become difficult to obtain due to the rapidly increasing demand for the vaccine development and export restriction. The development of suitable alternative assessment method is therefore recognized as a pressing global issue. With this in mind, we are working on developing method using human iPS cells and zebrafish eggs. For zebrafish eggs, 6 domestic institutions participated in this project and finalized the draft standardization protocol. As a result, high assay performance was obtained and will be published soon. Just for your information, the ISDT method far left, which is still in the research and development stage, is expected to be developed as an innovative evaluation method using human iPS cells, and the integral value of the amount of the various in gene expression induction by FGF, a key factor during development over time due to the chemical substances. Dr. Okubo, principal investigator of this project, will talk about it later, session 3. And next, as a part of evaluation of systemic toxicity, we are proposing several test methods for the assessment of immunotoxicity. We have proceeded with the development of the adverse outcome pathway, AOPs, and the detailed review paper, DRP, of immunosuppression. Through the development and validation of test methods based on these, we have developed the IL2-look assay as an OECD guideline. Ultimately, we aim to develop integrated approaches to testing and assessment IATA by integrating multiple test guidelines, guidance document, etc. In addition, as a part of immunotoxicity testing, Jack Baum has so far contributed to the development of guidelines for skin sensitization test method as a number of topical toxicity tests. Jack Baum has also been involved in the officialization of the monocyte activation test method as an in vitro method for pyrogen testing. We will continue to contribute to the development of IATA for the assessment of immunotoxicity by integrating existing multiple test guidelines, guidance document, AOPs, etc., and also by supporting the development of new AOPs based on our experience in the development of various test methods, and by supporting the development of new test methods based on these AOPs. As for toxicokinetics, Although toxicokinetics have become common knowledge in drug development, it has not necessarily been applied to the safety evaluation of general chemicals. But toxicokinetics is a key piece of information for predicting safety in humans, and the development of an affordable and reliable prediction system is considered essential for improving the prediction accuracy in non-animal test method. JACBAM is working to support the development of analytical method for toxicokinetics using MPS. And tomorrow afternoon, Dr. Yamasaki will introduce their effort on the MPS project, which is also one of the projects of Japan's Ministry of Economy, Trade and Industry, METI. JACBAM is also cooperating with other large-scale projects led by METI. One of these is development of MPS with the initial objective of supporting drug development. And now the project aims to industrial introduction of MPS and approval by regulatory authorities for its use in safety evaluation methods. The other is development of next-generation safety prediction method using artificial intelligence based on toxicity-related big data, AI SIPS project in short. They use some in vitro tests, including to predict toxicokinetics and animal database, and challenge in trying to apply for regulatory use on the safety evaluation of ordinary chemical substances. So we intended to continue to work to ensure the safety and security of the public, compete in the development of new test methods within the international community, and encourage domestic researchers. That's all for my talk. Thank you for listening.
Thank you very much, Dr. Yoko Hirabayashi. I shall open up to questions from the floor. Anyone has any question for Dr. Yoko? No? Yes.
Just a question on the hand look assay that was retracted, you say. Could you explain a bit more about that? And are there any lessons from that case to be learned?
Test method itself is not so bad, and maybe it could be go forward, but developer it takes time to establish the DRP, and developers unfortunately retired from the company, and they don't support anymore. That's the reason to withdraw the make the OECD guidelines. So if someone followed the method to develop guidelines, then maybe people are happy, but we cannot find out the supporter yet. Thank you.
Thank you. Any further questions? Okay, with that, thank you, Dr. Yoko. Thank you.
Thank you.
Yeah. Okay, the next speaker is Dr. Monique Bonhoeffer. He's from the European Chemicals Agency and a member of the Alternative Methods team of the Computational Assessment Unit. And part of his daily task is on the assessment of QSAR predictions and norms. He also works on the development and application of alternative methods, especially their validation, at the European Center of Validation of Alternative Methods. And he is also a faculty member at Johns Hopkins University. And with that, may I invite Dr. Monique to give us the presentation. Dr. Monique, please.
Thank you for the introduction, and thank you for the organizing for inviting me to discuss the regulatory landscape and critical needs for NAMs in the hazard assessment of industrial chemicals, and also giving me the opportunity to show you the efforts the European Chemicals Agency is making to step up for the promotion of alternative methods. So to structure our discussion today, I will start with the brief introduction to ECHA, and then I will walk you briefly through the EU chemical legislation and the current status and the REACH. And then I will talk about the role of NAMs in an animal testing-free regulatory system, and that was the subject of a NAM workshop that we organized beginning of June in Helsinki at the European Chemical Agency. And then I will talk about the use of NAMs and some examples of use of NAMs at ECHA. The European Commission adopted recently its chemical strategy for sustainability, where ECHA takes an active role in various areas. For example, banning the most harmful chemicals in consumer products, assessing how we introduce a mixture assessment factor in REACH, consolidating and simplifying chemical regulations, and Maurice showed you this morning a slide that I used to use, where you see all the implication of all the chemical pieces of legislation in Europe. Promoting alternatives to animal testing, which is the subject of today's talk. Boosting innovation and safe use by design of chemicals, and also the ambition to play a leading role globally. At the European Chemical Agency, we work for the safe use of chemicals, and our agency, among others, is responsible for implementing 4 big EU chemical regulations. So REACH, which stands for the registration, evaluation, authorization, and restriction of chemicals, and under this law, companies who make or import substances at certain tonnage over 1 ton per year need to register their chemicals with us, with ECHA. Under the classification and labeling and packaging regulation, companies need to make sure that their chemicals are classified, labeled, and packaged according to European rules. The biocidal products regulation makes sure that only approved substances and products are available for consumers. And the PIC, which is the prior informed consent regulation, is there to make sure that we have an overview of the import and export of hazardous chemicals to and from the EU. There are other tasks that ECHA, that the European Chemical Agency, is taking, like, for example, the directive on water, or the new regulation on batteries. But today we'll be talking mainly about REACH and CLP. I changed the slide from the one that Maurice used this morning, but we have in Europe an extensive chemical legislation for the protection of human health and the environment. And the regulatory system for industrial chemicals management relies on a horizontal generic approach based on the identification of hazard and properties of substances. REACH and CLP are the 2 key horizontal EU regulations that allow identification of hazard and the classification of substances. So basically, REACH and CLP are those 2 key horizontal European regulations. REACH defines what information we need to ensure safety. On the other hand, CLP shows how we use this information to manage the chemicals. These horizontal regulations are complemented with other sectorial legislation, like, for example, for pesticide, biocide, pharmaceuticals, or for worker safety legislation, environmental protection legislation, and product control legislation, for example, for detergent, toys, cosmetics, etc. It's not the purpose of my talk today to discuss all aspects of the legislation, but it is important to understand the key elements if you want to change and introduce NAM into this system. And I was talking during lunch about that, and about what level, for example, a researcher or developer needs to know about the regulation and the legislation to be able to introduce NAM in that system. We will not be talking about that, but it is important to understand the key elements if you want to change and introduce NAMs. And these are the fundamental elements that are highlighted here. So classification and labeling enables the identification of hazard or properties, and classification is based on adverse effects independently of exposure by applying agreed criteria, for example, global harmonized systems that are transported to CLP. CLP ensures that through harmonized classification and labeling, appropriate classification is made for most hazardous substances, and this measure can be enforced. And this system provides a framework for generic risk management. Generic approach means that when certain criteria are met, automatically management measures are applied automatically, not case by case. In this system, REACH feeds into CLP and ensure that industry provide adequate data. So REACH, this is where we provide the information, and this is where the standard information requirements are written, basically. REACH requires that data generated using standardized accepting testing methods, so for example, OECD test guidelines, enables the identification of adverse effects, and it compares with the GHS criteria, so we see the link between REACH and CLP, and enables the derivation of safety levels. Also, using OECD test guideline allows the mutual acceptance of data, which is important in terms of global regulation like REACH and CLP. So when it comes to the use of alternative under the REACH regulation, the best place to look at to have the information is the what we call the 117.3 report, which is this is an article of REACH that puts a legal obligation on the European Chemical Agency to report every 3 years on the status of implementation and use of non-animal test methods used in REACH registrations. We just published the 5th edition in June 2023, and that edition includes, if you are familiar with this report, and I think some of you are familiar with this report, we provided the data analysis of current REACH database as in all previous editions, so we could compare what is the evolution of the use and implementation of alternative methods. Additional to that, we made an analysis of just the newly registered substances for the last 3 years. And this is important because in 2018, the last registration deadline in REACH happened. So basically, everything in Europe that is in the market is registered, well, substances above 1 ton per year. And one thing that we included in the report this year is a discussion about ECHA's activities to promote NAMs, and also our contribution to the ongoing debate and transition towards full replacement of animal testing for industrial chemicals. So this is a report published by ECHA, and we're talking about transition toward full replacement of animal testing. It is just a discussion, but it is there. I think it is really, again, sharing the efforts we're making to step up for the promotion of alternative methods. I invite you to consult the reports, but I just give you some highlights here. So for example, when looking at the options used to fulfill the information requirement in the REACH registration database, this is the graph on the left side, we can see that adaptations continue to be used more than experimental studies. Adaptations are read-across, QSR, weight of evidence, and so on. Read-across continues also to be the most used adaptation in the REACH registrations. We're looking on the right side at the occurrence of studies over the years, over the last 3 years for those endpoints that have been amended in the annexes by introducing new requirements using in vitro methods. What we can see that the in vitro methods introduced recently are widely used for those three endpoints. We still have some in vivo data there, but in vitro is taken massively. So currently we are in the situation where replacing animal testing one-to-one is successful for some simple endpoints. I'm using the word simple here with caution. Nothing is really simple in this story. And we support this work through OECD work on defined approaches, for example. And we saw there, and it was mentioned already this morning, that this is a long process to happen. On the other hand, replacing animal testing one-to-one it's challenging for complex endpoints, again, complex, used cautiously. under the current regulatory framework, and this for many reasons. Standard information requirements currently refer to specific animal tests, very specific animal tests, and the alternative or the adaptations need to assume full equivalence to animal tests. The current system is regulated based on observed adversities, and the majority of the NAMs or new approach methods cannot directly predict adverse effects at systemic level this way. And we have also a lack of comprehensive knowledge in some cases. So this is basically the situation in which we attempt to introduce NAMs towards an animal-free regulatory system for industrial chemicals. And for this, we propose a way forward in 3 steps to close major gaps, to gain experience, and to propose a NAM fully compatible system, if we can, at the long term. In step 1, we would like to identify the critical needs to transit to an animal-free system to steer NAM development, and that was the subject of the NAM workshop I talked about. And while in step 2, we would like to apply already available NAMs under the current system, and we are collaborating with, we are supporting our colleagues at the European Commission to see how we can do that. And at the longer term, to rethink the overall system and see if we can have a system that is more compatible with NAMs that it is at the moment. These critical needs were described in the NAM workshop background document of the workshop that I just talked about. And here there are 3 of those critical needs that we identify. So hazard identification, which is the ability to demonstrate that NAMs can be used for a conclusive outcome on the hazard properties for a given regulatory endpoint. The hazard characterization, so how we use this molecular cellular level of information instead of the observed adversity, and extrapolation. And I'm glad that my colleague James from ASTAR talked about the TK just before me. So this workshop was organized by ECHA together with the European Commission, EPAA, which is the European Partnership for the Alternative Methods to Animal Testing, CEFIC, which is the Industry Council, and also NGOs PETA, to discuss the critical needs within the current regulatory system for bringing perspective from different stakeholders and to explore opportunities to increase the use of NAMs in the short and the long term. So the workshop summary report was just published today. So I didn't have the time to put a link there, but I invite you to read it. It's a very short report. It's not really very long, and I invite you to read it. It's just something like 15 pages, very, 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 very concise. But some key messages from the workshop is that we observed and we witnessed a strong commitment from all stakeholders to move towards that goal of animal-free chemical safety assessment, with clear goals and milestones to measure progress. Of course, there was some divergence on how this could be done for different regulatory contexts, and it was acknowledged that the regulatory context defines the readiness to apply NAMs. A targeted investment is required, and everybody in the workshop acknowledges that. And also, one thing that was mentioned many times, also even in the talks and presentations today, is there is a need of dialogue and collaboration between all stakeholders, across all sectors and geographical region. So now let's talk about the use of NAMs at ECHA and how we use them in our processes. So to support NAMs, we have 3 main pillars. So internal activities, which is the effective implementation of the integrated regulatory strategy. It is an ECHA process to identify and address risks of chemical concern. The second one is the investment and external collaboration. And the third one is making data available as much as we can. And I will show you an example of these three main pillars. So for the internal processes, we rely on the integrated regulatory strategy that connect the regulatory processes that we see on the right side, starting from how we receive the data, so registration, to how this data is evaluated through dossier evaluation or substance evaluation or PBT assessment, through the assessment of the regulatory needs. And the regulatory needs here means, is there a need for more data? Is there a need for further regulatory action or another legislation, or there is no risk? So those are the regulatory needs we are talking about. And to increase the pace and efficiency of identifying and addressing substances of concern, you can see that you have on the left side a process that is internal, which is grouping. And grouping here is used. We moved from examining substances one by one to grouping to expedite the assessment of those regulatory needs. Just to be clear, the main source of information here is still the ECHA database. However, external sources of prediction tools might be used to screen and assess consistency of groups for a given potential hazard. And this is where the NAM enter into play, and those are mainly QSR models and also high throughput and high content data that we use. A detailed description of the IRS report, you can see it, so you have a QR code there. And you can see also a description of how we use NAMs in the integrated regulatory strategy in the 117.3 report I was talking about. The second pillar is about the activities promoting alternatives. And our objectives there is really the international collaboration, scientific exploration jointly with research, consortia, academia, and also to support industry and promote harmonization. So I would like to emphasize here the number and variety of collaborator and collaboration we have. So on the left-hand side, you have the APCRA, which is accelerating the pace of chemical risk assessment, which is a government-to-government consortium, and you see that some of you here in the room are in this consortium. And there, the strategy is to use case studies that are valuable for all actors. And we are leading or working on two specific case studies. One is the retrospective case study where it shows the utility of in vitro bioavailability using, for example, ToxCast data. And there is a nice paper authored by Katie Paul Friedman from the EPA and also colleagues from ECHA on that. We are pursuing this one with a prospective case study where instead of using already existing data, prospectively data is generated. The second one is research consortia within Europe, and the flagship is ASPIS, which is a consortium of 3 research projects that was funded under Horizon Europe. And those are Precision Tox, OnTox, and Risk Hunter, and we are collaborating with them at a cluster level. The third example I would like to show for this external activities we are having is the collaboration with the industry. And those are just really few examples I'm showing here. And I would like to show the collaboration with CEFIC on a ring trial called Matching, where 6 different laboratories used metabolomics for grouping, and we helped them design the study and also analyze the data and ensure that the blind condition of the study were respected. And the third pillar is making data available. We facilitate the access to non-confidential data in Euclid formats, and Euclid really here is the flagship product, and you can see other products that we are co-funding or co-managing, like the QSR Toolbox, the eChem portal, and so on. And we have data available for download, so we have all the study results that are available for download, and those are data for registration of about 23,000 substances with information on physical properties, ecotox, fate, and tox information. And we have some, let's say, smaller collaboration that we had. So for example, we used Euclid to have about 348 approved pharmaceutical in Euclid format, and this is also available for download, and also some industry data that was provided by the European Federation of Pharmaceutical Industries and Association. So just some final remark. ECHA is really proactive to promote NAMs, and our activities in this respect are going far beyond the regulatory implementation of the current regulation. NAMs can be utilized now to refine, reduce, and replace animal testing under the current system, but this is not sufficient. And one other thing is that we need to be aware of the regulatory context to transition to an animal test-free system. Case studies can demonstrate that sharing data can accelerate this transition, but we need to have this awareness. And the last thing is that it is really collaborative efforts that requires buy-in by all stakeholders, regulators, industry, researchers, but also the public. And thank you for your attention.
Thank you. Thank you, Dr. Monique. Shall I open up questions from the floor? Yes, Gonzalo.
Thank you. And I suppose that this question just reflects my ignorance of the REACH registration process, but I'm very curious. So when a new sponsor brings a chemical, wants to bring a chemical to market in the EU space and approaches you, and you're charged to doing an hazard assessment of the chemical, do you need to do an hazard assessment just in absolute terms or in the context of the intended use of the chemical as outlined by the sponsor? Because related to this is, till what concentrations do you challenge your in vitro NAM systems? How does that, the circumstances inform the NAM evaluation?
So for the users, there are sectorial legislation that's, for example, if you're having in mind cosmetics, so there will be cosmetics regulation that will deal with that use. REACH is a horizontal legislation, so all chemicals before being put in the market needs to be registered, and there are very, very prescriptive information requirements that the registrants need to fulfill, depending on different different criteria. Those criteria, the first of them is the tonnage band. So the more the substance is made or important into Europe, the more information you will be required to provide to the agency. So this is how it works.
Okay. So perhaps to clarify a little bit better my question, so the hazard evaluation is done during the REACH registration or later on by a secondary entity that deals with specific use? Okay, I see. Sorry.
Yeah, so there is the registration, and there are internal processes that are transparently, published. So first, there will be, if you want to know about the technicalities of it. So first, there is a completeness check. Is, do you have all the information you need for that substance, for that tonnage, and so on? And then there is a dossier evaluation, which is basically a compliance check of what you have in the dossier. And then the risk assessor will go through all the endpoints and will look at the information that is provided. And you will do that by endpoint, so you will do that by hazard. So while you're doing that in compliance check, so we'll have some standard information requirement that could be adapted or waived, or that could be used to trigger additional studies. So this is how you do the process. After that, you will have member states you will have a decision about the compliance of your dossier. And then you will have, or it's compliant, or there is more need of more information, more data, or
Okay, all right, okay, thank you. Appreciate it.
Thank you.
Thank you very much, yeah.
Thank you. Okay, I think we need to move on to the next speaker. Thank you, Dr. Monique. Okay, may I invite our last speaker, who is Dr. Anna Lanzoni. So she's from EFSA, and she's involved in peer reviews of pesticide from 2013 to 2021, and she was involved also in the risk assessment of GMO at the GMO unit. And before that, she worked for 20 years as a toxicological pathologist at GSK, and is involved in preclinical regulatory studies and other projects development of new medicine. And without further ado, may I invite Dr. Anna Lanzoni for the presentation. Dr. Anna, please.
Thank you very much for the introduction, and thank you also for the invitation. What I would like to share with you today is the current EFSA activity, or activities actually, on investigating possible new approaches and tools for investigating the safety of protein. I wish to tell you that I'm going to present this now, but Antonio Fernandez, another colleague from EFSA, actually, has collaborated with me in the preparation of this. Okay, this. Okay. Sorry, but is this one? Yes, yes,
yeah,
now
that's
it
great.
does. I have to press more. Thank you. So why talking about safety of proteins and assessment of safety of the proteins in food and feed? There are many dimensions when we talk about proteins and proteins in food and feed. Of course, the nutritional side is the most, I can say, immediate we can think of, but there are many other possible uses, applications, intended, I mean, but also the presence, for example, of proteins in contaminants in food and feed. When I talk about intentional use, a couple of examples that to show here. For example, proteins are used as enzymes in food and feed technology, but also proteins can be used as pesticides in the pesticide content. Okay, so why, and now back. Okay, why to assess protein safety? Okay, proteins actually are part of our normal diet, and we are exposed to proteins by different ways, but proteins can also be toxic or pathogenic, actually. And here you can find an example on this side. You can talk about agglutinin, neurotoxins, and so on, from many sources to which we can be exposed. On the other side, a very big part of the story is also related to the possibility of proteins to induce immune-mediated reactions, which for commodity I'm going to use the term allergenicity, but I'm going to tackle a bit more in detail. By allergenicity, actually, as a common term we use in EFSA guidance as well, we mean different types of immune-mediated situation, including those mediated by IgE and those non-IgE mediated, typically celiac disease. Okay. So, I mentioned you that proteins are also part of the assessment that EFSA is conducting. How? A big part of that is done for proteins that are intended to be used. I mentioned you enzymes before, but we can also think about GMOs expressing new proteins, for example. And in this case, a pre-market assessment is required, and EFSA is supporting in the safety assessment of the proteins in that context. There are sectorial regulations supporting that, and also EFSA-specific guidance document. An example, a list is there. The overall target is indeed to offer the assessment which is relevant for consumers, but also for operators, workers, and also for the environment. Okay. So, why are we exploring new ways to assess the safety of proteins? Currently, proteins, as I told you, are assessed according to specific guidance and under specific sectorial regulation, according to a different set of tools and methodology that more or less are the same across the different areas. So for sure, bioinformatics plays a relevant role in there, but also molecular biology, as well as the use of in vivo studies and of in vitro studies. So as you can see, NAMs already are there and are embedded in several regulations and guidance. Of course, other information is needed and relevant, such as the history of safe use. Roughly speaking, the assessment is focusing on looking at information and testing the source, for example, of the proteins, but also the protein as such. However, there are some issues. Okay, it's just slow, okay. I can say that indeed there are recurrent issues. I used to work in the GMO, now I work in a pesticides area, and I see that the recurrent issues is that the main one is that the assessment of proteins is essentially mirroring, it has been built mirroring the chemical risk assessments. Many aspects, of course, can be nicely adapted, but some others not. Proteins are large molecules, and practical things such as administering by gavage to, in rodent studies, for example, a preparation of protein is not so simple and offers many uncertainty. And then you can think about allergenicity and in general all immune-mediated situations where animal models are definitely not the best option to progress. So at the same time, we are facing since a while evolution in protein science. Broadly speaking, there are many new data, actually, new repositories, new tools, and new approaches that we can think of. Of course, we try to have a look to the regulatory applicability and relevance of this, but we are trying to investigate in that direction. And so this actually is also important. There is a sort of pushing towards a more I'd say streamlined and fitting for purpose and strong assessment of proteins, driven, for example, by the under the umbrella of the European Green Deal. You can see a couple of branches which are definitely of interest. Novel food sources are really consider a way forward in the context of the Green Deal, as well as the sustainable use of pesticides that among the group of low-risk plant protection products mentioned, for example, microorganisms and their products such as proteins. So a strong push for the future to have stronger tools to afford possibly increase demand for assessing proteins. Okay, so what are we exploring? Two main branches, toxicity and allergenicity, as I mentioned you before. Toxicity is a newborn, I would say, in this context, as compared to allergenicity, where much more work has been done. Nevertheless, I will start from toxicity, and what I will show you are our work on exploring in silico in vitro tools to support the assessment. While for allergenicity later on, some examples. I want to mention that we are still in an exploratory phase. We are looking for new tools, new approaches, and new methodologies, and we are definitely trying to build the knowledge on that. Of course, regulatory relevance is under our eye and in our mind always, but I think it is still somehow premature to go in that direction. We need indeed to work. Toxicity. In silico prediction, preliminary information and results. First of all, it is a multi-annual project that has been put in place by EFSA, and for the time being, we are at step number 1. Step number 1 is actually exploring literature, let's say, broadly speaking, in order to develop an in silico approach to possibly predict protein toxicity. The next step, if this is going to be successful, will be could be a software development, and in end some EFSA guidance to support that. Step number 1 has been awarded, a procurement has been awarded by the University of Milan. The key steps of this activity are four, actually, starting from a literature search, scanning all the available information on prediction tools already available for predicting toxicity of proteins, setting up a protein benchmark data set, and we will see more than one, testing the tools identified in the literature actually against these protein data sets, and then setting a pipeline to predict toxicity. An important point I'd like to share with you, if coming, yes, okay, is that most of this work is based on public available information, which are present in UniProt KB, which was chosen by a previous contractor, Palazzolo et al. 2020, to support a previous preparatory EFSA activity on that. This is important because the target is to get away from very specific and maybe proprietary information and moving on to something which is public available. So based on UniProt, also the definition of toxic proteins was defined, and based on a gene ontology. Details can be found in Palazzolo et al. Where are we now? The literature has been explored. These are the tools that are available and that can be used to predict toxicity of some groups of proteins, as you can see there, with some limitations. Indeed, many others were identified but found not suitable. The second step actually was to create a protein benchmark data set, and in the end we resulted in three data sets, those considered true positives, true negatives, and those which are expected to be false negatives. The true positives are those which are non-toxic protein according to the definition and their classification in UniProt actually. And as you can see, the contractor identifies subgroups that you can find there. True negatives are those which have high sequence similarity to the toxic proteins, but they are not labeled as toxic. And the third group as those which are expected not to be correctly classified by the tool, since falling outside the specific scope of the tool. The results are there. These are preliminary, actually, so I just show you quickly those. Testing. As I told you, the third step was to test the tools against the identified benchmark. The results are not super exciting because both sensitivity and specificity are not considered the best. So the next step is by the contractor is to investigate hidden Markov models and blasting strategy. Work is ongoing on that. As regards the in vitro strategy, instead, we started again with another procurement, which was awarded to a consortium answers list. This is really started definitely more recently. The nice picture is that there is a nice dialogue between this contractor and the other contractor in order to make use, if possible, of the same protein data sets and possibly expand. And as I mentioned here, this has a more biological angle, if you like. They are really looking hard into adverse outcome pathways with the target to identify key elements that could be used for identifying suitable available in vitro tools to test a protein which ideally has been predicted as toxic or not. I go quickly because I think the time is not. I would like to pass to allergenicity, which is definitely more, not more mature, but definitely more rich. Okay. Yes, if you wish, in this scientific opinion by the GMO panel, all what I'm going to say is summarized in a nice way. I'd like to start with allergenicity per se, meaning by that IgE-mediated reaction. There is a procurement outside aiming to identify novel strategy, and the focus of that actually is really to focus on the ranking method and screening of potential allergens. I show you what I mean. So the inspiration came from some work actually that tried to plot the allergenicity potential, if you like, of different foods as derived from investigation from the clinics, from humans actually. In this plot you can see different foods from apple to sesame, for example, where you can see their potency and also the prevalence in percentage in a human population. Since the starting point is that indeed there will not be a 100% certain assessment of allergenicity, actually the idea is anyhow to try to make use of this information and to try to identify the main allergens in this food. This could allow then to make use of this ranking of foods in terms of potency and prevalence and translate this into individual allergens. I show you quickly. Yes, the overall idea then would be indeed to build up a sort of staggered approach where the first thing is really to have a fit-for-purpose databases of allergens, link to foods known to be allergenic, and then, according to this, try to categorize from no risk to high risk with some gradient inside, and then tailor ad hoc different testing strategies according to the degree of risk, to the low, medium, or high risk. This actually is indeed ongoing, and the contractor that I mentioned you before is indeed working on building up the information and identify the proper information to structure the strategy. As regards in vitro protein digestion, I will go very quickly. EFSA has started with this contract, actually, that gave very good result, testing in vitro different conditions and improving the possibility to investigate the digestibility of proteins. Work is still needed, but you can find very nice information in this preliminary work. Okay. I like very much this because, as you can see, this applies to IgE, situation raised by proteins. You can see that the top clinical relevance is there, followed by in silico, in vitro, and other investigation. In the context of allergenicity, we are a bit forced because, as I told you, animal models are not the best, but the large amount of clinical data could indeed support in that direction. Of course, a lot has to be done once preliminary bricks have been collected. Okay. Celiac disease, very, very quickly, just to show you that you might know very well what it is. Celiac disease is a non-IgE mediated uncontrolled condition, very well known, actually, in different steps. It does apply to a subset of the population expressing specific molecules recognizing the gluten peptide. Actually, EFSA has already developed a strategy which is in the revised version of the guidance on allergenicity assessment that starts indeed from collecting all the possible knowledge on protein, exposure, source, and whatever, searching for identity to specific sequence which are relevant and present in gluten and gluten-like proteins, and moving forward to some modeling in order to identify the risk possible, and then moving to in vitro testing if necessary. What I want to tell you is that EFSA has also initiated the procurement and also concluded, as far as I understood from Antonio, in order to release a software that is able to actually predict the possible binding of the protein, novel protein under investigation, to the specific receptor relevant in the pathogenesis of the celiac. Last bit, another activity, celiac disease is also under consideration in the context of another contract for building up an AOP, considering that all the different steps are very well known, and so this is another step that is going to be undertaken soon. So, again, also for celiac, yes. The idea is indeed to have possibly a database of clinical relevant celiac toxic T-cell epitopes, and then proceed to a ranking and define later on a strategy according to the low, medium, or high risk, moving then into the second phase as needed. So with that, I wish to thank you. I'm sorry for being a bit longer. These are the colleagues internal in EFSA working on this, but also I wish to thank all the contractors and all the experts, mainly from the GMO panel, have worked on that. Thank you very much.
Thank you. Thank you. Okay, may I just, maybe just one question before we break for tea break? Any questions for Dr. Anna? Yes, Bill. Over there.
You can hear me.
Yeah.
It's a very nice presentation, and I just wanted to ask, of course, we learned earlier today the importance of pharmacokinetics, metabolism, distribution of an agent in terms of toxicity. So I'm just wondering, in terms of proteins, is the dose response and metabolism as important as it is for traditional chemicals, just as a comparison?
I would say that in the context of food and feed safety, not yet. Not at all. We are talking about oral ingestion of new proteins in novel food, for example, but you can also think about dermal exposure by pesticides, for example. So not yet. The in vitro testing that we are investigating is a first step in that direction. Try to understand, at least via dietary ingestion, what happens to the protein. And it is not obvious. It is particularly important because the first site is the gut, where also allergenicity or celiac can start, indeed. But there is a lot to learn still, because it is not obvious that proteins are digested down to single amino acids. And small fragments, 9 peptide is sufficient to elicit the celiac disease. So not yet, but we are working towards. Thank you.
Yeah, thank you, Dr. Nanzoni. Thank you. Okay. We shall break for tea break. Be back here at
Thanks,
4:10, if possible. Yeah, thank you.
Thanks,
Thank you.
partner.
Thank you.